In this video, we'll talk about the nature of the genetic code, namely that 3 bases give us 1 amino acid, that each of these codons are unambiguous, that the genetic code has a degenerate or redundant nature, and that it's non-overlapping. We'll talk about what these things actually mean and how this can be evidenced from a given example. Okay, but before we do that, we're just going to review protein synthesis to remind ourselves of that process, and then we'll go into how the genetic code fits into that story. Remember that the whole point of transcription and translation is to produce a functional protein. That protein's only functional because it's got the right 3-dimensional shape. And the idea is that the 3-dimensional shape is correct because the order of the amino acids in the polypeptide chain is correct. And that order of amino acids is determined by the order of bases in DNA. So we'll just quickly review the process by which the amino acids are put in the correct order. All begins with transcription. Transcription involved a region of DNA corresponding to one gene, unwinding, and 2 strands separating from each other, allowing RNA polymerase to synthesize a complementary mRNA molecule. That mRNA molecule essentially takes the information from that section of the DNA out into the cytoplasm where it binds a ribosome. During translation, mRNA binds the ribosome. tRNA molecules with specific amino acids attached to each one bring amino acids to the ribosome, and if they are complementary to the codon sequence of 3 bases on the mRNA molecules that are being read at the time, they slot into position due to complementary base pairing again, and deliver a particular amino acid according to the codons being read on the mRNA. So when this codon is UCU, the complementary anticodon on tRNA has to be AGA. So only the tRNA that is bringing serine is then coming into this position at this time according to this codon. And serine is then the second amino acid in the chain. Accordingly, the next codon is UGG, and because of that, the only tRNA that can come into this position is the one bearing the anticodon ACC. And all the tRNAs that have ACC as their anticodon only carry TRP as their amino acid. So tryptophan is then added as the 3rd amino acid. Peptide bond is formed between these 2 amino acids, and the 3rd amino acid in that polypeptide chain is tryptophan. And so I hope you can see that the order of these amino acids is completely due to the order of the codons in the mRNA. And where did the sequence of bases in this mRNA come from? Completely complementary to the sequence of bases in the DNA. So the sequence of bases in the DNA determined the sequence of bases in mRNA, and the sequence of bases in mRNA is determining then the sequence of amino acids in the polypeptide chain, which, if they are correct, will allow the protein to fold up as it's supposed to and carry out the function that it's supposed to. That is protein synthesis. Transcription followed by translation, followed by a functioning protein. The gene is expressed and the protein goes on to add a particular functionality to the cells or tissues. That's protein synthesis. The next thing we have to discuss is the nature of the genetic code, so let's move on to that. These diagrams relate the particular amino acid and the codons that code for that particular amino acid. Remember, the codons ultimately determined by the sequence of bases in DNA. So let's take a particular example here and work with that for the rest of this tutorial. So let's say we had a gene sequence, okay, and our gene sequence was TAC, AGA, ACC, and AGT. I've made this up. Obviously, genes are much longer than that, many more bases. Okay, so this is our gene sequence, and the first thing you need to know about gene sequences is obviously, you know, in DNA you've got two strands, so we're going to have a complementary strand here which is going to read A, T, G, T, C, T, T, G, G, T, C, A. Okay, and these connected by hydrogen bonds, complementary base pairing, etc., etc. But you know, this video is not about the structure of DNA. Anyway, this is our gene, for example. Yeah, clearly the gene's going to carry on that way, as is it going to carry on that way. But this is our little bit that we're going to work with for an example. Okay, so that's our gene sequence. Now, the strand that is actually copied, so the strand which RNA polymerase uses to make the complementary RNA molecule called mRNA, that is called the template strand. Okay, so we'll just label that template. So this right here, that is called the template strand because that is the strand that is going to be used by RNA polymerase to make the mRNA molecule. So because the mRNA is going to be complementary to this, as is this strand right here, this strand is called the coding strand because it will very much resemble the sequence of bases in the mRNA molecule, only difference being instead of T's, the mRNA will have U's. So, you know, that might be a good way to get a shortcut to transcribe a strand. Okay, so we will then label that. This is called the coding strand. Okay. Other descriptors of the two strands are also sense and antisense. So the strand right here, because it reads pretty much as the mRNA molecule, it's also called the sense strand because it makes sense. Okay, so I will call that sense strand. The template strand, therefore, being called the antisense strand also. Okay, so coding, I guess the template strand then also becomes the non-coding strand. So the template strand, antisense strand, coding strand, sense. So when transcription happens, our template strand is going to be used to make a complementary RNA strand, and it's going to read very much like our coding strand or the sense strand, the complementary strand to the template strand. So our mRNA that RNA polymerase will be making during transcription is going to read with every T replaced with U, A, U, G, U, C, U, U, G, G, So that's the mRNA molecule that is produced when our template strand on our gene sequence is transcribed. Then the mRNA is translated. And when the mRNA is translated, as we discussed earlier, the sequence of bases here is then used to put the amino acids in the correct order. And that's really where our genetic code comes into play, because it's here that our sequence of bases is used to determine which amino acids are put together in the polypeptide chain. So, as we can see from the genetic code, that it's the sequence of 3 bases that gives rise to a particular amino acid, and we saw that earlier, that it's sequences of 3 bases on the tRNA, the anticodon, that bind with their complementary codon on the mRNA to determine which amino acid is going to be added to the polypeptide chain there. So that's the next thing that we're going to label here, because it's the sequences of 3 bases that determine amino acids. And the sequence of 3 bases on the mRNA that determines which particular amino acid is going to be added to the chain is called the codon. And then relating to that, we're just going to add an annotation back on the DNA molecule, that it's the sequence on the DNA strand that gives rise to our codon is called the triplet. So it's the triplet here that gives rise to our codon on the mRNA, triplet here that gives rise to our codon on the mRNA, and that is what determines which amino acid is going to be in the polypeptide. So the flow of information is really DNA, mRNA, and then polypeptide and protein. Okay, so this is where we use our genetic code now. So let's say we're using this diagram up here. We could equally just use this one. But our first codon is AUG, first letter A, second letter U, so second letter up here, first letter here, second letter here, and third letter we have to just pick from this, so I'll show you. So first letter A, second letter U, so we're looking in this box right here, and then within this box our third letter is G, so UCA is this one that we are reading. The last one, AUG, gives us met or methionine. So when the codon reads AUG, the amino acid that will always be added as a consequence of that codon is going to be methionine, met. So let's just separate our codons there. Okay. So moving on then. Our next codon is UCU. So U, second letter C, so it's this box that we're looking in, and then our final letter is U, and so it's the first one that we're looking at. UCU gives us serine. So UCU gives us serine. And we'll use this one just for fun this time. So UGG, first one U, second one G, third one G, TRP, tryptophan. And finally we have UCA, so U, C, and A gives us serine again. So our hypothetical gene sequence of this gives us a polypeptide or primary structure of methionine, serine, tryptophan, and serine. So that's from going from here to here is transcription, from here to here is translation, and the use of the genetic code really came into play during translation. Okay, so this is the flow of information. Now what we're going to do is we're going to take this example here and we're going to use that to look at the nature of the genetic code. Now we are really discussing the nature of the genetic code. So what does this tell us about that? So first thing is, because we have, so we've got 1, 2, 3, 4, we've got 3 times 4, we've got 12 bases, and 12 bases give us 1, 2, 3, 4 amino acids. So what does that tell us about the genetic code? First is that 3 bases give us 1 amino acid. So every 3 bases is determining 1 amino acid. So that's the first thing about the genetic code. Okay, and remember that these 3 bases on the mRNA are called codons, on the DNA they're called triplets. 2. Second one being that there could well have been a scenario where you read the first 3 bases, TAC, or let's say AUG, then the next 3 bases, instead of starting from where the first 3 finished, so instead of the next codon starting UCU, the next codon could well be, after AUG, we could read UGU, and then the one after that might be GUC. A little bit confusing, I know, but in a hypothetical situation, it could easily have been that way. AUG followed by UGU followed by GUC followed by UCU. So it's not that way though. You read the first 3 bases AUG, and then the next 3 bases are read after that, UCU. Codons do not overlap with each other. So the next very important thing about the genetic code is that these codons are not overlapping with each other. One codon finishes, then the next one is read after that. This is when you hear about the genetic code being non-overlapping, this is what it's referring to. It's how the codons are read. So, second point being that the genetic code is non-overlapping. Okay, one codon ends, then the next codon is read after that. So the next 3 bases are the next codon. 3. Third point being, very important, that each codon is unambiguous. Each codon results in a particular amino acid, and there's no room for any codon being interpreted as one amino acid or another one. So let's pick an example here. So let's look at our particular example here. If we have AUG, AUG can only be methionine. UCU, now you might say, well, yeah, okay, but methionine only comes up once. Okay, so let's take UCU then. Okay, now, serine has a number of codons, so a number of different codons can give rise to serine, but UCU, let's just take UCU. UCU, right there, only results in serine. UCU doesn't give rise to any other amino acid apart from serine. So that means that the code is unambiguous. UCU means serine and nothing else. Similarly, any other codon only gives rise to that particular amino acid or means that particular thing. It doesn't, there's no leeway for interpreting a codon as any other thing than what it's meant to code for. This means that the code is unambiguous. Okay, that's really important. What isn't? 4. Last thing is, you may have heard the code being referred to as either redundant or degenerate. And what that's referring to is the fact that whereas each codon only gives rise to one amino acid, there can be more than one codon that can give rise to a particular amino acid. So let's go back to our particular example right here. Serine, UCU gives rise to serine, but UCA also gives rise to serine. You see? So because there can be more than one codon that gives us serine, that is referred to as the genetic code being redundant or degenerate. So that's what it's referring to. Do not mistake this with being ambiguous. Okay? So that the unambiguousness is referring to any given codon only gives rise to one amino acid. Okay? But when you look at the amino acids and you look in the opposite direction, if you look at a particular amino acid, so threonine right here, there's a number of different codons that can give rise to threonine. And this is what's being referred to as redundancy or being degenerate. Okay? So, I mean, maybe we can just make a note of that. The unambiguousness is referring to codons giving rise to amino acids, but the redundant nature is really, if you think about amino acids and the codons that are able to give rise to them. Okay, so that's that. And you know, this last one has important implications because, just make a note, you know, some mutations, so like point mutations or single base changes, let's just call that single base changes. So in terms of mutations, if you have single base changes, where one base is swapped for another one, or mutates and the sequence changes and gives rise to another base, there is the possibility that the new codon produced also gives rise to the same amino acid. And so that particular mutation might not affect the structure of the protein and its function. So there's that important application of that last point right there. But essentially that's the genetic code as you need to know it at this level. I might do another video, but this is kind of long-term, looking at all, you know, the evidence, the experimental evidence that allowed us to know this about the genetic code, but that's maybe down the line a little bit. Right now I'm focusing on kind of, the things needed for the exams. So I hope you found this useful. Please do give feedback in the comment section below, and if you have any further questions on the genetic code, again, use the comments. I will get back to you. Thank you very much, and good luck, guys.